Alright, so today we're looking at proteins. Starting off with proteins, we know that proteins are made up of tiny molecules called amino acids. Now, amino acids are linked together. A lot of them linked together form a chain, and that complex chain we call proteins. Now, so let us start with the structure of an amino acid. So an amino acid has this general structure. The general structure of the amino acid, as you can see right there, has two parts to it and a central carbon. So the central carbon is the carbon in the middle right here, and the R group is a variable group. It might be listed in other material, other textbook as variable group. And to the sides you have the carboxyl group and the amino group. Amino group is that N and two hydrogens bonded to it. So the variable group, sometimes listed as a side chain, we just refer to it as the R group. Your carboxyl group can be written as this, or it can be written as C double bond O with the OH, right? Either of these is acceptable. Good. All right, so now we're looking at a bond between two amino acid groups. So note, this time I have the carboxyl group written as C double bond O and bonded to a OH. Please note, remember from the first time we start, the carbon has only 1, 2, 3, 4 valence electrons, so it will only participate in 4 bonds. So look, it's the same thing over here. And remember, this reaction is a condensation reaction. So condensation reaction leads to the removal of a water molecule. Alright, so when we remove the water molecule, this is the bond that is formed now. Oh, and there is an error on the diagram as I look here, because nitrogen only have 1, 2, 3 valence electrons, so this should not be here. That was an error. Good. So 3 valence, 1, 2, 3. Over here, 1, 2, 3. This bond that is formed now is called a peptide bond. Good. When we have now the molecule itself is going to be called a dipeptide, because it is a bond. It is two amino acid molecules bonded together. When we have many peptide bonds, that give us a polypeptide, and polypeptides is what we call proteins. So just to review that, that's the formation of the peptide bond is between the carboxyl group and the amino group, where we remove a water molecule and form a peptide bond between the carbon and the nitrogen molecule of the two amino acids, and we said that this leads to a dipeptide molecule. Many peptide bonds, which means that you have a chain of these continuously, gives us polypeptide molecules. All right, now we're going to look at protein structure. So please note that the amino acids are the only structure, are the basic units then of protein molecules. Not like in the carbohydrates where you have alpha glucose, beta glucose, and fructose, and so on. Here in proteins, we're dealing with amino acids alone. Now, proteins are molecules. They do not remain the same. They change after a while depending on their chain. So we have 4 structures, or 4 levels in which we describe protein structure. So the first level is that we have the primary structure proteins. Primary structure proteins are just a chain or sequence of amino acids, and this is determined by the genetic code or the gene, which is just a length of DNA. So just like how I show you the polypeptide is just a chain of amino acids, right? This is a linear thing, so you just have this chain going along with the polypeptides bonded like here. So you'd have many amino acids bonded in a linear fashion. That would give you your primary structure. One more thing to note in reference to the primary structure is that you have many different amino acids. Good. And the identity of the amino acid is really determined by the R group. Good. We'll get further into that as we continue, as we progress, but suffices just to say is just that each amino acid is just not exactly the same. You have different types of amino acid. As a matter of fact, we have basically 20 different types of amino acid. So when we talk about the primary structure being a chain or sequence of amino acids determined by the length of DNA, it means that we have different amino acids lined up together depending on what the DNA says. Okay, now when it comes to the secondary structure, secondary structure is just referencing the fact that the primary structure don't remain linear in nature. It will actually start to coil into different shapes. So some part of the chain will coil into a alpha helix and other parts may coil into a beta pleated sheet, and the coil and the pleated sheet is held together by hydrogen bonds. Cool. So alpha helix would be like this helical shape right here, reminds you of amylose from yesterday's class. Beta pleated sheet is kind of something looking like this. Good. So you will have some parts of the chain coil like that, the alpha helix, and some parts like the beta pleated sheet.